Imagine this: tiny bacteria, those microscopic organisms we often battle in our daily lives, aren't just randomly clinging to surfaces. They're actually feeling them out, responding to the physical world in ways that shape their entire existence. This fascinating discovery could revolutionize how we combat infections and design smarter materials. But wait, this isn't just about bacteria. It's about unraveling the hidden forces that drive their behavior. And trust me, it's the part most people miss that makes it truly groundbreaking. In the real world, bacteria don't often swim freely like lonely fish in an ocean. Instead, they form sticky communities called biofilms, attaching to everything from medical implants to your smartphone screen or even the tissues inside our bodies. Their actions, how they stick, multiply, cluster, or secrete glue-like substances to hold their biofilm together, are all influenced by the mechanical push and pull they experience when interacting with these surfaces. For beginners, think of it like how you might adjust your grip on a slippery object versus a rough one. Bacteria sense these interactions through their cell membranes, the protective barriers around their tiny forms. Why does this matter? Grasping how bacteria interact with surfaces is crucial for fighting the stubborn buildup of biofilms, which can cause serious health issues like infections in hospitals. It also paves the way for inventing new antibacterial materials and improving technologies that detect biological signals. Yet, despite its importance, the basic physical processes behind these interactions have remained largely a mystery. The way bacteria perceive and react to surfaces can deeply impact their biology, from growth patterns to survival strategies. Enter a team of scientists, spearheaded by Dr. Cristina Flores at IMDEA Nanociencia in Madrid, Spain, who've cracked open this enigma. They've pioneered a cutting-edge technique to actually see and measure the mechanical stress, or tension, in bacterial membranes when they touch various surfaces. Their findings are detailed in a paper published in the Proceedings of the National Academy of Sciences, offering a fresh perspective on these microbial mechanics. At the heart of their method is fluorescence lifetime imaging microscopy, FLIM, combined with a specially designed fluorescent probe called FLIPR-TR. This tool allows them to detect even the slightest shifts in membrane tension in living bacteria as they engage with different materials. It's like having a high-tech camera that captures the invisible forces at the bacterial surface boundary, providing a clear view into the physical dynamics at play. So, how does FLIPR-TR work? This commercially available fluorescent marker is engineered to zero in on cell membranes, and its fluorescence lifetime, essentially how long it glows before fading, shifts based on changes in tension. Made up of two dithionothiophenol molecules, the probe flattens out under pressure within the membrane and twists back when tension eases, altering its glow time accordingly. For context, imagine a flexible toy that changes shape under squeeze. FLIPR-TR does something similar, turning mechanical cues into measurable light signals. Previously, FLIPR-TR was primarily used in studies of mammalian cells, like those in human tissues, with limited success in bacteria due to technical hurdles. But now, the IMDEA Nanociencia researchers have shown it's perfectly suited for bacterial models. What's more, it's precise enough to tell apart interactions with surfaces of varying stickiness and textures. Their experiments prove that FLIPR-TR effectively labels both gram-positive bacteria, think Staphylococcus, with thick protective walls, and gram-negative ones, like E. coli, featuring a thinner outer layer, and it finely distinguishes how their membranes react to diverse surfaces and nanoscale structures. By analyzing differences in fluorescence lifetime, they can uncover unique attachment strategies and even spot membranes stretching on nanostructured surfaces designed to kill bacteria mechanically. Think tiny spikes that puncture them on contact. This research paves the path to deeper insights into how physical signals steer bacterial life, and it offers blueprints for crafting biointeractive materials. These could either repel bacterial adhesion, helping prevent infections, or encourage it for beneficial uses in biotechnology. Applications might include safer medical devices, advanced biosensors, or innovative materials that promote good bacteria while fending off harmful ones. But here's where it gets controversial. As we develop surfaces that actively kill bacteria, are we risking the evolution of super-resistant strains, much like overuse of antibiotics has done? And this is the part most people miss. Could manipulating bacterial feelings lead to unintended consequences in ecosystems, like disrupting natural microbial balances? It's a double-edged sword, balancing human health against broader environmental impacts. What do you think? Should we embrace these mechano-bactericidal designs as a powerful weapon against infections, or are there ethical lines we're crossing by engineering surfaces to fight back? Do you believe this could spark a new era of antibiotic-free treatments, or might it create more problems than it solves? Share your opinions in the comments. I'm curious to hear your take. For more details, check out the full study by M. Carmen Gonzalez-Garcia et al., titled Exploring Bacteria or Surface Interactions with a Fluorescent Membrane Tension Probe, published in Proceedings of the National Academy of Sciences, 2025. DOI 10.1073 NARS.2512977122. Available at https://dx.doi.org/1073/pnars.2512977122. Citation: How Bacteria Feel Surfaces. Fluorescent Probe Visualizes and Quantifies Membrane Tension. 2025, November 18th. Retrieved the 18th of November 2025 from https://physics.org/news/bacteria-surfaces-fluorescent-probe-visualizes.html. This document is subject to copyright. Apart from any fair dealing for the purpose of private study or research, no part may be reproduced without the written permission. The content is provided for information purposes only.